Hello student, I am here to discussing the southern blotting technique. This technique is also called as a hybridization technique. This technique is used for analyzing the related genes in the DNA. So the DNA which is present in the chromosome, and on this DNA there are lot of the genes are there. So these genes we have studied by the southern blotting technique. Particularly the southern blotting technique is only applicable for DNA. So this technique is developed by scientist E.M. Southern in 1975. This technique gives a physical map of gene located on the chromosome. In this technique, first we have to isolate the DNA, and this DNA should be digested. So here this figure shows we have to isolate this DNA, that is a DNA sample. This DNA should digested with the restriction enzyme. Then this restriction enzyme cut this DNA into the fragment and we will get fragmented DNA. So many double stranded fragmented DNA will be formed or developed by this restriction enzyme. This double-stranded DNA should be denatured by using alkali such as NaOH. You have to add the NaOH. Therefore, the DNA becomes single-stranded. The single-stranded DNA are of various size, and because the restriction site on this DNA are different, and therefore different sized DNA fragments are developed during this procedure. So here there is a necessity to separate the single stranded DNA fragment, and therefore we have to separate the DNA by using electrophoresis technique. So in electrophoresis, so if you see the actually the kit, so this is the electrophoresis kit. So in this kit, this is the vial or the tube. So this tube, we pour the solution of the DNA fragment into this vial, and then this kit has anode, that is a plus charge, and this is a cathode, that is a minus charge. When we pour the DNA fragment solution in this vial or in this tube, then the smaller DNA fragment immediately, here we can see, immediately go toward the positive charge of agar gel. So here there is agar gel plate, and on this agar gel, this DNA, which is smaller DNA, they are immediately go toward the plus positive, we call it charge, and the largest DNA or larger DNA, they are behind this smaller DNA. So this is we call it as a electrophoresis. Then DNA has you have know the DNA has the negative charge, and therefore it always attract toward the positive charge, that is the anode. So here we have complete the electrophoresis. So the next one, so first here we have to again go back. So here we will get, this is the plate, agar gel plate, and on this agar gel plate we will get, this is the DNA fragment. So in next we have to prepare the standard kit. Here we have to prepare the standard kit, and this kit, first we have to take the broad glass jar. So here this is a broad glass jar, and inside this glass jar you have to keep the glass plate. This glass plate, on this glass plate you have to put the filter paper. So normal filter paper, the two margin, so here this is the one margin, this is another, two margin are folded in such a way that the folded portion should be deep into the buffer solution and absorb the buffer. So here In this jar we have to add the buffer solution. You know the buffer solution is the solution which have no change the pH. And on this buffer solution there is a filter paper, and on filter paper we have to put the agar gel which have the DNA fragment, and on this gel we have to put the this black colored, that is nitrocellulose membrane paper. So this nitrocellulose membrane paper is the paper which actually not allow the DNA molecule. That means here it is not filterable the DNA molecule. Around this nitrocellulose membrane you have to keep the bunch of the filter paper. We call it as a filter paper towel, and then kept the weight on this filter paper towel. So here this is the kit. So in this kit the filter paper, so here this folded paper margin of the folded paper absorb the buffer solution. So here the buffer solution is absorbed by this buffer filter paper, and then this solution, buffer solution, is goes toward the upward because the gel is there. So this gel, agar gel, also absorb the buffer. Then again nitrocellulose, ultimately this filter paper, it absorb the buffer solution. But one thing is that during the movement of this buffer to upward direction, or during absorption of the buffer solution by this filter paper or by this nitrocellulose membrane paper or filter paper, the DNA is stuck to the lower side of this nitrocellulose membrane paper, and the DNA cannot pass this nitrocellulose membrane paper, and this filter paper towel absorb the complete this buffer solution. So here we have we will get the DNA which are stuck toward the lower side of this nitrocellulose membrane filter paper or membrane paper. Then you have to remove the nitrocellulose membrane, the labeled DNA. So one more thing here. So here this is the nitrocellulose membrane paper. So on this nitrocellulose membrane paper we have to release the DNA probe. So here labeled DNA probe you have to release it for hybridization, and that means here this there are so many fragments of DNA. We have released some DNA probe. So this DNA probe are nothing but the special DNA, or which have very small nucleotide or small number of the nucleotide, and this nucleotide is labeled, or it may be it is bounded by certain enzyme or certain fluorescent dye. That means this is the DNA probe which we have artificially bounded by different enzymes or fluorescent, or it may be labeled by isotopic element. So the sequence of this probe we have known. When we have to release this DNA probe, then the DNA which is complementary with this DNA probe, they are joined together. That means the DNA probe is joined, or it is when it is complementary, then it is joined. So here we have shown only one. This is a DNA probe. So this DNA green color DNA probe is complementary with this fragment, and therefore it is joined with this DNA. That means both this DNA structure which we will get, we call it as a hybridization. Why it is hybridization? Because this green colored nucleotide is the probe which is actually prepared, and this small red colored, this is the DNA fragment which we have take from the DNA sample. So here this is called as a hybridization. But finally, final step, that is the autoradiography. After washing the nitrocellulose membrane paper, we go the X-ray film which have the different bands of the double standard. So here this double standard band, or this double standard DNA, we will get on the autoradiography film. So here we show this autoradiography film. So here this is the band. So here this is the complementary DNA. we will get such a complementary DNA, that is the hybridized DNA. So we have know the sequence of nucleotide of the probe, then we can easily identify the DNA or the sequence of the nucleotide present on this fragment of DNA. So here in this technique we have to blot the DNA on nitrocellulose membrane, or we have blot this DNA on the autoradiography. That means here this southern blotting technique is used to analyze only the DNA. I think you have got what is meant by southern